Today I am here to discuss mode of action of antineoplastic agents. First category is alkylating agents. The alkylating agents are a class of drugs that are capable of forming covalent bonds with important biomolecules. The major targets of drug action are nucleophilic groups present on DNA, especially the 7th position of guanine. Alkylation of DNA is thought to lead to cell death. Potential mechanism of cell death include activation of apoptosis caused by p53 activation and disruption of template function of DNA. Here, apoptosis represents the death of cells which occurs as a normal and controlled part of an organism's growth or development, while p53 is also known as TP53 or tumor protein. It is a gene that codes for a protein that regulates the cell cycle and hence functions as tumor suppression. It is very important for cells in multicellular organisms to suppress cancer. In this picture, you can see double-strand DNA structure. Alkylating agents binds on the guanine residue at 7th position. They causes interstrand linking and cross-linking. This is the mechanism of alkylation by alkylating agents. Alkylating agent binds to DNA at 7th position of guanine residue and they causes alkylation of DNA, which results in interstrand linking and cross-linking. Now we will elaborate this mechanism. Alkylating agents form highly reactive carbonium ion. This carbonium ion transfer alkyl groups to nucleophilic sites on DNA bases, which results in cross-linking, abnormal base pairing, and DNA strand breakage. DNA strand breakage causes decreased cell proliferation. Decreased cell proliferation represents reduction in expansion rate of a cell population by cell division. Alkylation also damages RNA and proteins. This one is normal cell cycle. The DNA alkylating agents are not cell cycle specific, but they are more toxic to cells in late G1 and S phase. This is the time DNA is unwinding and exposing its nucleotides, enhancing the chance that vulnerable DNA functional groups will encounter the electrophilic attack that leads to its own destruction. So the concluded mode of action is that alkylating agents forms highly reactive carbonium ions. These carbonium ions transfer alkyl groups to nucleophilic sites on DNA bases, which results in cross-linking, abnormal base pairing, and DNA strand breakage. These all factors results in destruction of abnormal DNA. Next category is antimetabolites. Antimetabolites are structurally related to normal compounds that exist within the cell. They generally interfere with the availability of normal purine or pyrimidine nucleotide precursors, either by inhibiting their synthesis or by competing with them in DNA or RNA synthesis. Their maximal cytotoxic effects are in S phase, and therefore they are cell cycle specific. In this category, we will discuss mode of action of folate antagonists. As we all know, folic acid is an essential dietary factor. It is converted by enzymatic reduction to series of tetrahydrofolate cofactors that provide carbon groups for the synthesis of precursors of DNA and RNA. Precursors of DNA include thymidylate and purines, and precursors of RNA includes purines. First of all, folic acid is converted into dihydrofolate, which is further converted into tetrahydrofolate in the presence of dihydrofolate reductase. Tetrahydrofolate converts into 5,10 methylene tetrahydrofolate. Furthermore, 5,10 methylene tetrahydrofolate is converted into 10 formyl tetrahydrofolate, which results in de novo purine synthesis, where the term de novo represents new purine synthesis. Folate antagonist methotrexate inhibits the enzyme dihydrofolate reductase. Inhibition of dihydrofolate reductase leads to partial depletion of tetrahydrofolate cofactors 5,10 methylene tetrahydrofolic acid and 10-formyl tetrahydrofolic acid. These cofactors are essential for the respective synthesis of thiamidylate and purines. In addition, methotrexate undergoes conversion to a series of polyglutamates in both normal and tumor cells, which inhibits the enzyme thiamidylate synthase. Next subcategory of antimetabolites is of purine antagonists. Purine antagonists 6-mercaptopurine and 6-thioguanine are converted in the body to corresponding mono and ribonucleotides, which inhibit the conversion of inosine monophosphate to adenine and guanine nucleotides. Adenine and guanine nucleotides are the building blocks for RNA and DNA. There is also feedback inhibition of de novo purine synthesis. Next subcategory of antimetabolites includes pyrimidine antagonists. Pyrimidine antagonist 5-fluorouracil is converted in the body to the corresponding 5-fluoro-2-deoxyuridine monophosphate. This 5-fluoro-2-deoxyuridine monophosphate inhibits thiamidylate synthase and blocks the conversion of deoxyuridylic acid to deoxythiamidyllic acid. This deoxythiamidyllic acid is also known as thymidine monophosphate, and it is denoted by DTMP. It is a nucleotide that is used as a monomer in DNA. Inhibition of this conversion leads to elective failure of DNA synthesis. Mode of action of antibiotics as antineoplastic agents. Doxorubicin is capable of causing breaks in DNA strands by activating topoisomerase II and generating quinine-type free radicals. Topoisomerase II is an essential enzyme that relaxes DNA supercoiling during replication and transcription. Doxorubicin interacts with DNA by intercalation and inhibition of macromolecular biosynthesis. This inhibits the progression of enzyme topoisomerase II, which relaxes supercoils in DNA for transcription and then stops the process of replication. Transcription is the first step in gene expression, in which information from a gene is used to construct a functional product such as protein. The goal of transcription is to make an RNA copy of a gene's DNA sequence. DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. DNA replication occurs in all living organisms, acting as the most essential part for biological inheritance. Bleomycin is a mixture of closely related glycopeptides, antibiotics. It acts through binding to DNA, which results in single and double strand breaks following free radical formation and inhibition of DNA synthesis. It chelates copper or iron between DNA strands and causes chain scission and inhibit repair. The DNA fragmentation is due to oxidation of a DNA bleomycin Fe2 positive complex to DNA bleomycin Fe3 positive complex. In this conversion, electrons are liberated, which reacts with oxygen to form radicals which cause strand breakage. Last is mode of action of natural products as antineoplastic agents. Epipodophylotoxins. Etoposide blocks cell in late SG2 phase of the cell cycle through inhibition of topoisomerase II. It inhibits DNA synthesis by forming a complex with topoisomerase II and DNA. This complex induces breaks in double-strand DNA and prevents repair by topoisomerase II binding. Accumulated breaks in DNA prevent entry into the mitotic phase of cell division and lead to cell death. Microtubule inhibitor. Vinca alkaloids cause mitotic arrest by promoting the dissolution of microtubules in the cells. These drugs arrest cells in metaphase and interfere with the synthesis of transfer RNA, possibly by acting as acylating agents. Metaphase is a stage during the process of cell division, and microtubules are the key component of cytoskeleton of eukaryotic cells, which are formed by the polymerization of a dimer of two globular proteins, alpha and beta tubulin. So by inhibiting the ability of cancer cells to divide by acting upon tubulin, they prevent it from forming into microtubules, a necessary component for cellular division. Thank you.